Welcome back to Dead Reckoning, the Human Life Expectancy channel. This is the first of several videos where we'll focus on perhaps the main advance in science, technology, and medicine that has the potential to drive increased human life expectancy, namely our new capability to read and write DNA, the code of life. Genetic technologies offer the potential to identify and predict a range of conditions, revolutionize human reproduction, treat many diseases, and even the possibility of curing aging itself, resulting in a profound increase in human life expectancy. Since DNA is the code of life, learning to read it and the epigenetic switches that turn it on or off, and learning to edit and rewrite the genome will profoundly affect us all. This video focuses on how genetic and epigenetic tests were developed and how they might extend HLE. Future videos will focus on using genetic technologies to revolutionize human reproduction and engineer organisms going forward. So, how did we learn to read the human genome? We had to answer two basic questions. One, what letters do we read? And two, how do we read them? To the first question, by the early 1920s, biochemists knew that nucleic acids came in two forms, DNA and RNA. DNA is the code of life, whereas RNA is a messenger carrying instructions from DNA for controlling the synthesis of proteins. This established what's known today as the circular flow of genetic information, where genes encode RNAs in a process known as transcription. RNA then builds proteins in a process known as translation. Proteins form and regulate organisms. Organisms sense environments, and environments influence proteins in RNA. And then that regulates the genes, and so on and so on. It's found that DNA is organized in sections called chromosomes that usually come in pairs. In humans, DNA is divided into 23 pairs of chromosomes, 46 in all. On April 25, 1953, James Watson, Francis Crick, Rosalind Franklin, and Maurice Wilkins published Molecular Structure of Nucleic Acids, which revealed the double helix structure of DNA, and in 1962 they shared the Nobel Prize for this work, except for Rosalind Franklin, who had since passed away. They showed that both DNA and RNA were long chains made of 4 nucleic bases strung together on a helix backbone made of alternating sugar and phosphate groups. The 4 bases are adenine, thymine, cytosine, and guanine, abbreviated A, T, C, and G. Since A's almost always pair with T's, and C's almost always pair with G's, in cell replication, single strands of DNA unzip and find new complementary nucleotides. In RNA, the thymine is switched to uracil, or U. The human genome consists of 3.2 billion base pairs, about as many letters as in 700 large books. Just as the number and order of letters determine the meaning of a book, the number and order of these base pairs determines the myriad attributes of an organism. Mathematically, it only takes about 130 base pair combinations to equal the number of estimated particles in the observable universe. Since there's 3.2 billion base pairs in human DNA, we shouldn't be surprised at its ability to guide all cells and life forms. The complete book of letters, the complete sequence of DNA, is referred to as the genome, and genes can be thought of as paragraphs within that book. Random errors in copying genes are called mutations. Sometimes the mutations don't matter at all. Either they result in no change to the organism, no phenotypic change, as geneticists call it, or a change that has no impact on survival. But other times the mutations can matter a lot. As with words, sometimes small changes in letters or other groupings change everything. For example, only a few minor changes turns kids exchange into kid sex change, something really quite different. And only a few minor changes turns theirs into the IRS. So you get the idea. Sometimes the mutations result in phenotypic changes that do materially improve or reduce an organism's ability to survive and reproduce, driving natural selection and evolution. At birth, an average of 50 to 100 of the 6.4 billion letters that make up your DNA are different from your parents. While this is a very small number, over millennia, this random mutation is enough to cause the evolution of our species. If we could read the order of A, T, C, and Gs, or sequence them, as geneticists call it, we could read the book of DNA. So how can we read the sequence of the genome? In the mid-1970s, Frederick Sanger and Alan Coulson from Cambridge made significant progress in being able to sequence genes. They discovered that they could use special bases that would cause the cell's genome to break up into fragments, then stain and electrically sort the fragments by size, and use a special camera to read genetic patterns. This is referred to as first-generation genome sequencing, or Sanger sequencing. Then, in 1983, biochemist Kary Mullis conceived of the polymerase chain reaction, PCR, process, which uses thermal cycling and a special protein from an underwater extremophile bacteria to rapidly make millions to billions of copies of DNA samples, allowing scientists to take a very small sample of DNA and amplify it to be large enough to study in detail. PCR quickly became a fundamental tool enabling genetic sequencing. Driven by Moore's Law and PCR, in April 2003, the Human Genome Project was declared complete. It took over 13 years at a cost of over $2.7 billion to sequence one representative set of all parts of each human chromosome and identified over 20,000 genes that make up the parts of the human body. In the 2000s, a new form of sequencing called next-generation sequencing, or NGS, caused the costs of sequencing to fall even more rapidly than the exponential reduction from Moore's Law. NGS is an enormous scale-up of the former standard Sanger sequencing method. Human DNA is split into tens of thousands of fragments and sequenced at the same time, and the genome is reassembled using computers to find the overlaps between the sequences. This made computers and computer science a critical part of genetic science and greatly reduced the time required for sequencing. Today it costs only about $700 to sequence a full gene, and relevant parts can be sequenced for under $200. Lower processing costs set off a race to sequence genomes, and more DNA was sequenced than ever. As exciting as the sequencing of the human genome was, it proved to be a bit anticlimactic. As more and more DNA was sequenced, it was discovered that many people carry genes that never get expressed. For example, having the gene for breast cancer, yet never developing the disease. Geneticists started to look around the genome for switches that might turn on or turn off the expression of certain genes, and the science of epigenetics was born. Epi means above, over, or on in Latin. If the genome is the parts list of life, the epigenome is the assembly instructions, showing which parts to use in what order. It turns out that our bodies are even more fantastic than we had thought. Not only do our genes evolve generation by generation through homologous recombination, but our epigenomes change within our lifetimes to meet the needs of life in different environments and at different stages of aging. Since the epigenome has been shown to be an important driver of aging and life expectancy, we need to read it. And to do so, we must answer the same two questions we answered for reading the genome. What can we read, and how can we read it? To answer the first question, we need to briefly dip back into biology. Since the late 1600s, it's been known that embryos start as single cells that divide and differentiate into the many organs and tissue types necessary to form living organisms. But how do individual cells know how and when to differentiate? By the 1920s, it was known that when an embryonic cell divides, its daughter cells both receive a replica of its DNA. Since the genetic code, or DNA, of all cells are the same, the cells must express the genes differently to differentiate into the many different types of cells that make up the human body. In the 1950s, Conrad Waddington, an English embryologist, tried to understand what environmental factors might cause differences in gene expression. Waddington likened embryonic cell differentiation to thousands of marbles rolling down a sloping landscape full of crags, nooks, and crevices. He proposed that as every cell traced its unique path down the Waddington landscape, It's settled into some particular channel or crag, causing it to express certain genes and differentiate into a specific type of cell. He also defined the science of epigenetics as the study of DNA for switches that control gene expression. Epigenetics, Waddington wrote, concerns, quote, the interaction of genes with their environment that brings their phenotype into being, unquote. It was discovered that DNA strands are wound around spools of protein called histones, and both DNA and histones can have various chemical handles, cranks, and levers attached to them to help turn genes on or off. These handles, cranks, or levers comprise your epigenome. Numerous studies have confirmed that epigenetic signatures can predict how long an organism might live, what behaviors they've exhibited, and many other factors. In the late 1970s, scientists identified DNA methylation as one of the major switches governing gene expression. In DNA methylation, enzymes in our cells attach a methyl group to a cytosine located next to a guanine in our DNA, like charms on a bracelet. They act as powerful switches that can turn on, turn off, amplify, or mute the DNA in individual cells. So, to read an epigenetic signature, we must know the location of these methyl groups. This answers the first question. As for the second question, how can we read it? With some fancy chemistry, we can actually use the same sequencing methods used to read the genome. In the early 2000s, it was discovered that a process called Bisulfite Conversion could be used to differentiate and detect unmethylated versus methylated cytosines. In short, treating DNA with bisulfite converts cytosine residues into another base called uracil, but leaves the methylated cytosines unaffected. So DNA that's been treated with bisulfite retains only methylated cytosines, which can then be read as part of the otherwise normal genome sequencing process. Comparing these to the cytosines on the original sequence, which did not involve bisulfite processes, reveals which CPGs or sites were originally methylated and unmethylated. Today, a range of companies provide genetic and epigenetic tests. Commercial genetic testing companies such as 23andMe and Ancestry.com process saliva and blood samples, use microarrays and polymerase chain reaction PCR-based solutions for genetic testing only. They don't do any epigenetics. While PCR-based methods are cost-effective and commercially scalable, they can only measure a limited number of sites along the genome. As a result, over the past 10 years, microarrays have been used for the bulk of research and commercial applications. Large companies have formed to provide microarrays and sequencing for genetics and epigenetics. With revenues of over $3 billion, Illumina is the market leader. Their CEO, Francis D'Souza, announced that the cost of sequencing genomes should soon be under $200. Another major provider, Beijing Genomics Institute, or BGI, has also committed to this same pricing. In addition to microarrays, nanopore technology offers the prospect of revolutionizing DNA sequencing once again. Oxford Nanopore's sequencing technology uses electricity to push DNA through holes in proteins to read its sequence. It offers real-time analysis for rapid insights in fully scalable formats from pocket to population scale that can analyze native DNA and RNA or sequence any length of fragment to achieve short to ultra-long read lengths. For epigenetics, the nanopore process can directly detect methylated cytosines without using bisulfite or a second sequencing pass. Another company called PacBio recently announced a highly accurate long-read sequencing, which it calls HiFi, that offers 99.9% accuracy. It can also identify methyl cytosines for epigenetic tests. Other new entrants in the sequencing space include Ultima Genomics and Element Biosciences, promising low prices and high accuracy. All of this makes us confident that the quality of genetic and epigenetic tests will continue to rise as costs continue to fall. So, what can genetic and epigenetic tests tell us? Plenty. To start, they provide predictors of life expectancy. In the past few years, biomarkers of aging based on DNA methylation, known as epigenetic clocks, have gotten better and better at predicting biological aging. In general, when your biological age is less than your chronological age, your life expectancy is higher than your chronological age would suggest. You're literally young for your age. In 2016, Steve Horvath published an important study on the epigenetic clock, which showed that DNA methylation markers can be used to predict biological age more precisely than chronological age. The study looked at the mortality of over 10,000 people tracked over a 10-year-plus period and developed an epigenetic predictor for all-cause mortality that did a significantly better job of predicting all-cause mortality than chronological age. How much better? The epigenetic predictor is about as important in explaining mortality as knowing whether or not somebody smokes, which from an actuarial perspective can be plus or minus 7 years. So this is a big factor. Since then, several other epigenetic predictors of biological age have also been identified that provide similar insights to Horvath's epigenetic clock, including pheno-age, grim-age, and deep-M-age. There are also non-epigenetic predictors of biological age that measure things that vary with age, such as eye scans, walking speed, and even sitting and rising tests. However, since studies have shown that epigenetic factors are actually a hallmark of aging instead of merely correlating with it, and can accelerate or slow the aging process directly, epigenetic tests are more reliable indicators of biological age. More on this in future videos. Epigenetic tests of biological age create the possibility for individuals, doctors, health insurers, and others to track an individual's health and longevity over time. This is a game changer. Throughout history, humans have sought strategies for increasing their lifespans, but we've lacked a metric of biological age. It's the equivalent of trying to diet without a weight scale. Now, epigenetic tests that predict biological age can be used to see which interventions slow or even reduce biological aging. This can be used to greatly accelerate the discovery of interventions that have the greatest impacts for different groups of people. Genome-wide association studies have shown that genetic and epigenetic markers can also be used as reliable predictors of multiple diseases, conditions, and behaviors. So-called polygenic risk scores, or PRSs, have been developed for a broad range of conditions, including cancer, diabetes, and pulmonary fibrosis, phenotypic variations such as obesity and blood pressure, and behaviors that impact health, including smoking tobacco and alcohol consumption. The extraordinary reality seems to be that our bodies respond to diseases and behaviors by switching on or off different genes to enhance our survival. When we smoke, get cancer, or experience some other medical shock, our epigenome basically kicks into gear and turns on and off a range of genes to help us to cope. Increasingly, epigenetic tests can pick up these responses, offering an important window into health and related behaviors. Similar to epigenetic tests of biological age, polygenic risk scores, PRSs, combine information from genetic and epigenetic tests with other information to assess an individual's disease and mortality risk going forward. They're used to complement existing risk factors to improve disease screening programs, regular screening for high-risk individuals, reduced and unnecessary screening for low-risk individuals. More precise screening leads to improved lifestyle or medical interventions to enhance HLE and the quality of life. PRSs combine information from large numbers of DNA variants that they've inherited, often hundreds to millions across the genome, to give a single numerical score representing an individual's propensity to develop specific diseases. PRSs are now available for some of the largest non-communicable diseases, including coronary artery disease, many types of cancer, type 2 diabetes, and many others. Recent studies even show that genetic and epigenetic tests can also be used to diagnose many psychological conditions. For example, genetic and epigenetic tests are used in liquid biopsies to diagnose cancers earlier, more reliably, and with less invasive procedures. Traditionally, when a patient is suspected of having cancer, a surgery called a medical biopsy is performed to obtain a sample of the potentially cancerous tissue that's then examined under the microscope for cancerous growth. This is slow and sometimes painful, and only works when the cancerous tissues are large enough to identify. Today, liquid biopsies are eliminating the need for this surgery and providing earlier, better results. Liquid biopsies screen blood samples for cancer cells from a tumor or for pieces of DNA from tumor cells. They're used to identify cancers before tumors have become large enough to identify. This is critically important because early detection is critical for curing cancer and many other diseases. It also allows individuals to take remediative actions, from seeking treatment and cures to taking preventative measures, eating differently or exercising more, to simply planning their lives more effectively. Sequencing company Illumina is now launching Galleri, the $1,250 liquid biopsy test from Grail, which it acquired in August of 2021. Grail's come up with a test for more than 50 cancers that detects altered methylation patterns in DNA circulating freely in the blood. So, how will genetic and epigenetic tests impact human life expectancy? When our team worked through the potential impacts of genetic and epigenetic testing on all the causes of death, we found that they could have a very significant impact on HLE within the next 20 years. They can be used to measure whether any proposed medical or anti-aging interventions are being successful by measuring the difference between biological age and the chronological age as various interventions are undertaken. This can increase the pace of discovery, leading to more saved lives and longer life expectancy. As we said, genetic and epigenetic tests can produce polygenic risk scores that can help to diagnose heart disease, cancer, and many other top killers earlier when they're much easier to cure, and to track the efficacy of treatments. They may also discourage smoking, alcohol, and other behaviors that contribute to heart disease, cancer, and many other major killers by making these behaviors more transparent, discoverable, and trackable. Also, driven by Moore's law and advancements in machine learning, we expect that the science of genetic and epigenetic testing will advance rapidly. In addition, the potential for this new testing technology to cause new deaths is extremely low, limited to the remote likelihood of people getting erroneous test results and making inappropriate health decisions. The main uncertainty on their impacts on global HLE is the rate and completeness of their adoption. Accordingly, we averaged multiple adoption scenarios and coming to our conclusion that global HLE could increase by more than 10 years in the next 20 years. With their enormous potential impacts on HLE and rapidly falling costs, genetic and epigenetic testing is poised to usher in a new age of healthcare. Increasingly, we'll be able to read the code of life, the switches that turn it on and off, and take actions that will improve our health and lengthen global HLE. In our next video, we'll see that genetic and epigenetic testing will also likely be used to revolutionize human reproduction. In the meantime, please hit the like button below, subscribe to the Dead Reckoning channel to make sure you don't miss our next videos, and post comments for the topics you'd like to have us cover. Until next time, as we say at Human Life Expectancy Inc., hi hua! Please, do what you can to help yourself and help us all to live longer.